Alright guys, this is Dr. Surya Singha with the Animal Diversity Lecture. We are continuing on. We finished talking about protostomes and now we are moving on to talk about animal diversity, their form and function with respect to deuterostomes. And as you can see in the cladogram, we are actually at the, you know, kind of at a very derived clade, and you can see over here you are looking at more basal clades, right? I intentionally flipped this cladogram so that I want you to understand that basal organisms are not always on the bottom orientation of a cladogram of a phylogenetic tree, right? It actually depends on um, the branch position as opposed to the lateral vertical position, right? So if you look at deuterostomes, they actually descends of multiple branches starting from the evolution of the most basal animal, right? It actually undergoes multiple branches, right? Over here after multicellularity, goes through bilateral ancestors, goes through bilateral symmetry, and then it goes through development of the coeloms, development of the central nervous system, development of civilization, right? So, and then of course you see evolution of deuterostomy, right? So as I just explained, deuterostomes have undergone multiple steps, have gone through multiple ancestors, right, from the multicellular all the way up to the one that actually first developed the central nervous system, civilization, and coelom development. So they are going to carry a lot of synapomorphic features that are unique to animals and some ancestral features that we observed in certain basal animals, right? And there are two groups that we are going to talk about among deuterostomes: echinodermata and chordata. Chordata includes us, echinodermata or echinodermata includes starfish and that particular group. Okay, moving on, deuterostomes, which includes echinodermata, are actually monophyletic because all the members of deuterostomes includes all the branches that is emerging from this particular deuterostomic ancestor that shows deuterostomic embryonic development. Now, deuterostomes are large-bodied, right? They actually relatively speaking, they have a larger body size compared to some of the protostomes, and they demonstrate complexity in terms of body organization, body structure. Um, Now, what makes a deuterostome? Now, we have to go back way back when we started first talking about protostomes. I did mention that this depends on what happens to the deuterostomy versus protostomy depends on what happens to the initial opening in the embryo when embryonic development proceeds, right? If that first opening becomes the mouth, you are a protostome, right? If the first opening um, in the embryo in the early embryo becomes mouth and becomes the anus and mouth develops secondarily, well, then you are looking at a deuterostome, right? So because of that, here's again a beautiful opportunity to use logic rather than rogue memorization. If mouth is produced secondarily and anus is produced first, the gut development of the digestive tract, digestive system starts from the posterior to the anterior end, right? It's not the way more intuitive to you. You might think, oh, it should happen from mouth to the anus, because if you remember among some protostomes, I mean even before protostome evolution, we had some animals who had a mouth but not an anus, right? So you would think, okay, then the mouth should be producing first in any animal if that is the basic way animals are evolving. But no, you're talking about deuterostomes, a completely different that actually follow completely different embryonic development, right? So it's kind of cool to think about this. For them, it's actually, for deuterostomes, it's not posterior to anterior, it is anterior to posterior, right? Because blastopore, the early opening in the embryo, becomes the anus. The cleavage, if you remember from the early lecture, is radial, where the cell division of the zygote happens in a radial pattern, meaning it starts from the center of the zygote and radiates outwards, right? Radiates outward. And in protostomes, it was spiral. Okay. And also, remember the coelom, the opening, the primary opening you see that is surrounded by, that is lined by mesoderm, right? So deuterostome have this coelom, right? You know, they inherited it from their previous pre-deuterostomic ancestors, and now that still develops as outpocketings or divergences from the mesoderm. Now, that's actually nothing new because we already talked about that. Um, Deuterostomes contains four phyla: echinoderms. You see them over here, which includes starfish, sea urchins, sea cucumbers. It's a really cool group of animals. Hemichordates. These guys, they are also known as acorn worms. They actually have a more like a burrowing, deposit feeding or a suspension feeding mode of life. Largely aquatic animals. And then Xenotabella, these guys, which are actually a really interesting group of animals. They are fairly recently discovered to science, and we are still learning quite a bit about them. And I'm not going to go into detail about them because we are actually, just covering the basics of animal kingdom. Last part, which is actually us, chordates. All right. And you can say again, if you look at the cladogram, right, chordates are the most derived, right, or recently evolved group of deuterostomes, right? All right, so first let's go by, piece by piece. Let's first talk about echinoderms. Echinoderms, the term echinoderm means, derm is, echino means spiny. So their skin has spines, right? So now this spininess is much more conspicuous, prominent, right? Or in certain groups of animals, but could be extremely reduced in other groups of animals. But most of them show this spininess or presence of sharp spikes coming out of their skin. They're marine, right? Exclusively marine phylum, and their presence is more remarkable in deep water, but not to mention that in certain shallow water coastal environments, you do see some echinoderms, particularly sea urchins, or starfish are not that uncommon in shallow water environments, but mostly they are found in deep water marine environments. Extremely diverse in terms of different species, as well as in the body plans. In fact, the body plans you observed in different classes within this phylum is pretty amazing. I'm showing examples of a sea urchin over here and a starfish over there, right? Because those are the groups that you must be already familiar with. So, what is the very basic fundamental echinoderm body plan look like? Synapomorphic features, right? Synapomorphic features, what kind of synapomorphic features can we identify in this particular group that help us to group all of those as a monophyletic clade? They have radial symmetry. This is really interesting because this is not the first time radial symmetry evolving in this in animal kingdom. We did observe that in certain cylinderates. Okay, and so this is a re-evolution of radial symmetry, right? But what happens to the bilateral symmetry? Now, this is actually again a good moment to use logic, because remember, echinoderms are descending from, not from radially symmetrical ancestors, they are descending from bilaterally symmetrical ancestors. So they are larvae, in fact, so they have larval stages. That's one point to remember. Second point is the larvae is bilaterally symmetrical, not radially symmetrical. So radial symmetry is a character you can only observe in certain adults, right? And it does not say that these guys are related to coelenterates. In fact, echinoderms are more closely related to us chordates than to cnidarians, right? Or than to cylinderates. Now, cylinderates is a kind of an older term we use for Cnidarians. I don't care which one you use, but anyways, don't mix up this, right? They're not related to those basal radial symmetrical animals, right? Really interesting point. um, these guys do not have a distinct head. Because they don't have distinct head, they can move at any direction, right? So an adult, right, with radial symmetry can move towards any of the directions they are, that is available to them. So it's also a really interesting point. Why? Because they are descending from an ancestor that actually had cephalization. So these guys secondarily lost cephalization, right? So this brings up a really cool point. Can a particular highly derived animal group lose a synapomorphic feature? Can they lose an ancestral feature? Absolutely yes. I mean, down the road when you take other courses, particularly I teach coded biology, I go into detail how this happened. It's extremely fascinating how biological evolution works. So for yes, they are descending from ancestors who had a head, right? But these guys have actually lost, secondarily lost that characteristic. They have a skeleton. The skeleton is an endoskeleton, internal skeleton, that's calcareous. What does that mean? Calcareous means a skeleton made out of calcium, like in you and me, right? And the exoskeleton in certain groups of echinoderms can be flexible, right? Meaning you will get joints in between different parts, so allowing some movements. But in certain cases, it's rigid, it's fused. The different parts of the skeletal system are actually more strongly fused, thus more rigid, structured, and inflexible. Okay. The purpose of having a skeleton is multiple, and you already know a few of them. One is protection, particularly if it is fused, if it is calcium based, it can provide protection to your body, right? Probably encircle more sensitive or more fragile organs and protect them. Secondarily, support. Remember, I mean, think about, apply this to any skeleton. Remember the first time we talk about a skeleton, who I elaborated about the skeleton was in terms of arthropods who had an external skeleton, and they actually supported the body, right? The support comes in two different ways. One, supporting to keep the body shape, body form, right? And that applies to any skeleton. And the secondary thing a skeleton can do is actually provide support for attachment of muscles. In the case of arthropods, that was evident, and it is evident now as well, right? So now again, why am I reflecting back? It's not to cause you more trouble, but actually kind of give you more opportunities to understand this subject better. So the point is, try to compare what you observe across different animals. That actually, helps you to remember these things, and it becomes more logic based rather than, like just blind memorization. All right. Water vascular system. This is the first time you are hearing about this particular word, water vascular system, right? It's also known as hydrovascular system. There's two different terms, and this water vascular system has another unique feature called tube feet. Now, this system is something you will study in detail in the lab. So in terms of, if you look at my examples, this is a diagram of what the water vascular system look like, and this is actually the skeleton of a starfish. This is a skeleton of a brittle star. So starfish's water vascular system provide kind of a water-based pressure that kind of helps to operate the entire water vascular system, particularly to move the tube feet part of the animal, right? If you look at over here, right, the water vascular system has multiple components. You have at the center, right, you have this ring canal, right? And then ring canal has something called madreporite. So madreporite has an opening, a hole over here. So water actually moves into the water vascular system through the madreporite, and madreporite has something called a stone canal, which actually brings that water to the ring canal, and the ring canal radiates into the limbs, right? And those radiating parts into the limbs called radial canal. Very easy to remember. Ring-like part, ring canal, right? Radiating parts, radial canal. And radial canals then feeds into this, what you call as a tube feed, okay? And those tube feeds are the ones that actually gets triggered by the water pressure created within the water vascular system that helps to move the tube feeds. Movement of tube feed is extremely important because it helps the entire animal to move. The entire animal to move, stay in place but move its different body parts, as well as to capture food. So the canal system, this water vascular system, are like a network of water pipes that actually distributes water to back and forth from the environment to the tube feeds. And the water that actually that enters through madreporite can also exit through madreporite. Okay, cool. So another thing that I want you to question yourself here, right, is actually, okay, wait a minute. These guys are descending from coelom, and we are seeing already some really cool shit happening, right? So what happens to the coelom? Do they have a coelom? Yes, they have a quite spacious coelom. It's an open fluid-filled body cavity lined with and containing different tissues and organs. So yeah, they do have a coelom, right? So what do you find within the coelomic cavity? Well, large gonads, right? And they have gonads, and then those gonads are quite large. In fact, the more mature the animal is, the larger the gonads are, right? And also, usually, they have a complete digestive system with a mouth and an anus. And they feed by turning their stomach inside out. So their mouth is actually usually located on the ventral side, usually, and you will understand why I say usually. And what they do is, through their mouth they extrude their stomach inside out and produce digestive enzymes, right? Do the digestion and then kind of takes back whatever they digest back into the stomach, and then insert the stomach into their own body. Kind of really cool if you think about it. Or maybe gross, but what I always say, guys, is gross stuff that animals do are pretty cool to a scientist. Echinoderms, with certain exceptions, they do not have an elaborate respiratory system, or do not have a respiratory system in general, with the exception of sea cucumbers. And I will explain why later on. They have a very rudimentary circulatory system, which I believe you might get to experience in your labs. The water vascular system that I just explained takes over some of the functions performed by circulatory system and respiratory system. That's why you don't see the development of these parts. And likewise, the nervous system and the sensory system are poorly developed in the echinoderms. Alrighty, let's continue on. So, how do echinoderms feed? There are some echinoderms are predators. For example, sea stars. What they use is they use their tube feed. So remember those tiny little peg-like things in the water vascular system, right? Let's go slightly backward. These things, right? So they use those to kind of pry open bivalves, right? Remember bivalves, those mollusks, right? What they do is they actually, with the water vascular system having a hydrostatic function, they can exert pressure on to the tube feed, which actually the pressure itself can actually forcefully open the two bivalve shelves. And after prying open the bivalve, these guys invert, right, their stomach from inside out through that opening in the bivalve shell, and then the digestive juices are produced into the bivalve, and the poor helpless bivalve undergoes digestion, and then the resulting and then what happens when you have digestive enzymes on a poor bivalve flesh? It starts digesting the bivalve flesh, and then the digested components are now absorbed back into the stomach, and then the stomach goes back into the starfish's body, right? How cool is that? Sounds like an alien movie. I believe, I'm pretty sure, like it's been a while since I actually watched a movie like this alien movie series actually has something like that. There we kind of extrude their stomach um, for the purpose of digestion. It's a really cool thing. I mean, me myself being a movie buff and a movie geek or sci-fi movie geek, any of these, like weird sci-fi movies with an alien part or some sort of a mutant, that actually has been inspired by some reality that happens in the animal kingdom, right? Well, of course, they take that reality to the next level to make it more artistic. It's really cool, when you know science, what kind of cool stuff you can do with it. Okay, back to the lecture. Suspension feeding is another mechanism how echinoderms feed. So in this case, they extend their tube feet out into the water, and they use tube feet to sort of flick food particles that is suspended in the water column, right? And then that suspended food particle are actually then channeled towards cilia that actually is present in their body, and this movement of cilia channels those food particles towards their mouth. Deposit feeding, very similar to suspension feeding. In this case, what they do is they use their tube feet, those tiny feet, to secrete mucus, right? And then what they do is they kind of soak up the food material on their substrate, right, with the mucus, and then they move the mucus that contains food into their mouth by kind of moving their tube feet around. Again, try to remember the difference between suspension feeding and deposit feeding. They are very similar, right? Suspension feeding is actually, both are form of filter feeding, first. Second, suspension feeding is actually when you filter food particles from water columns. Deposit feeding is when you are filtering or collecting food particles from a solid substrate. Okay, that's the actual difference. Now, I remember what I always tell you, for all of the lectures I do, for all the animals I cover, there is videos. Please do go watch these videos, right? And on your own. If it is harder to understand without me present, drop by the office hours. I will explain what's going on in these videos. I find it usually unnecessary for me to explain the video in the class. If we were meeting in person, I would have gone over them, but they are cool and self-explanatory. Please do go watch them in order to understand these materials better. It will, trust me when I say this, watch them. It helps a lot, like particularly with respect to this tube feet. Watch it. It's kind of is creepy in an interesting way to watch how tube feet move. The tube feet's the way I look at, it's like a worm attached to an animal, right? To these echinoderms that actually, helps them to do stuff really cool, right? So there are several classes within echinoderms, right? Sea star, asteroids, brittle star, ophiuroids, sea lilies, right? Looks like a plant. Look at that guy, right? Looks like a plant. Crinoidea. They belong to the group Crinoidea. Echinoidea. Now, don't mistake this. Echinoidea sounds very much like Echinodermata, right? Echinoidea includes sand dollars and sea stars, right? And then sea cucumbers includes Holtheroidea, right? So look at how different their body plans are, although they belong to the same function, same phylum. First, let's talk about asteroids, the sea stars. All right. So they live on hard or soft substrates, right? Hard substrates like rocky intertidal shores. Soft substrates like sandy beaches or sandy bottoms of marine deep water environments, right? So, like I said, yes, they actually are found in coastal environments, right? So, they do have 5 or more long arms radiating from the central region of their body. So in fact, when I say 5 or more, I can actually be a little bit precise and say 5 or multiples of 5, right? It's like 5, it could be 5 arms, 10, 15, like that, right? You might mostly know of those with 5 arms, but there are other asteroids that actually have multiples of 5. Um, the central region of the body contains the mouth, right? The anus, mouth and anus on the opposite sides, mouth on the bottom, anus on the top, and the stomach, all located within that central part, right? And and their sexes are separate. They have male individuals, female individuals, producing sperms and eggs. Um, Sexual reproduction predominates to the large part, which actually occurs through fertilization of eggs with sperms. And the reproductive organs are not in the central disc, they are actually in the arms. In fact, relate this to when you look into your dissections in your labs, because you actually will get to see some of the sea stars where their entire limb is filled with nothing but gonads, right? That actually happens quite a lot. It's really interesting to see that happening. I mean, imagine what is the primary function of an animal, right? Everything they do, eating, growing, finding the best habitat, all of them is to ensure successful reproduction. What best ways you can do it other than investing everything you have to produce large gonads? Larger the gonads, more reproductive cells you produce. That is, that argument is as simple as that. So the fertilization is largely external, where eggs and sperms meet in the water. But there are few instances where that happens internally. They produce a very large number of gametes, and the larvae that results out of the fertilization could have a longer lifespan, and like most other larvae, they are planktonic, right? And another cool thing about these guys, about asteroids in particular, but can be generalized to most echinoderms, is their ability to regenerate lost limbs, right? You can actually take a sea star, chop off its limb, and it will grow back. And in fact, in some of the videos, right, if you look, if you search for this, you will find some photographs or even some videos of sea stars with one short limb and multiple long limbs. Why is that? It's not a malformation, it's actually an instant where a sea star is growing a lost limb. It's really important, because if something were to predate on this animal, or if it was to get physically damaged by some other means, or were amputated because of a disease or something like that, right, like a wasting disease or something like that, these guys can regenerate it completely, right? So if you see a shorter arm and multiple longer arms, you are not looking at a mutant, you're actually looking at a sea star that is regenerating its lost limb, right? It's remarkable. Tell you what happens if you cut off two limbs? Will they regenerate? Yes. Well, what if you cut off all the limbs, just leave the central disc behind? Still, they will regenerate all the 5 limbs. It's just a matter of time, right? It's extremely cool. And this is extremely important to the pharmaceuticals and the medical sector, because we, as mammals, have completely lost our ability to regenerate lost limbs, right? If you lose a limb, you lose it forever. But if we can kind of understand how we lost it, it's why it is not, why we have lost that ability. Well, chances are that we actually have the genome for the regeneration. Remember who we are descending from. We are deuterostomes. We are not that far apart. Our ancestor as mammals are not that far apart from our ancestor from deuterostomes. So is it because we actually have the genome for limb generation, but that genome is not, we cannot turn on that genome, right? Is there a way where we can actually like understand that genome that is responsible for limb regeneration or limb regrowth? Can we kind of splice it into humans? If exploring these kind of things is fascinating. I haven't read a whole lot about that, but particularly not with respect to asteroids, but I have read some literature where they studied that amphibians actually having the ability to regenerate their limbs. Interestingly enough, the amphibians' ability to regenerate limbs are more prominent in their larval stages. less prominent in adult stages. And some adult amphibians cannot regenerate lost limbs, but larvae, the tadpoles, can. Um, so that also provides evidence that the ability to regenerate limbs, the genome, or rather the genetic backbone for that, is not completely lost. Chances are it's turned off um, among vertebrates. Chances are it's turned off among mammals, right? So it's really interesting if we can uncover how to turn it on or to splice it if we happen to completely lose that part. Anyways, it's really interesting stuff. Let me tell you one last interesting medical important stuff about these guys. They have a really unique form of cells called coelomocytes, which is kind of an amoeba-like cells that can move like an amoeba within the coelom, within the body cavity. That actually kind of functionally resembles blood cells of vertebrates, right? Particularly, their function seem to be is capture and destroy any foreign bodies like bacteria and virus, right? So that is also medically relevant because that could be kind of the turning point of evolution of mammalian or vertebrate immune system, right? I mean, back in the college, I read a very early days, like a very early paper that describes this really weird cells produced by echinoderms, particularly starfish, that is able to perform some general immune functions. A lot of cool stuff I can go on talking about these critters. Now let's move into the next group, Echinoidea, sand dollars, and sea urchins. So this is sand dollar, right? And this is a sea urchin. Sand dollar is called a sand dollar because it actually looks like a dollar, right? It's flat, dorsoventrally compressed. In sea urchins, there is some dorsoventral compression, but they still maintain their globular body form. So sea urchins, they still have the globe-shaped body structure, body form, and then they have a lot of long spines, and they do have tube feet. Using these tube feet and partly using those spines, and those spines are movable, they can crawl along the substrate, right? Now, if you look at the sand dollars, they are much more flattened, dorsoventrally compressed. So there is more like disc shaped. That's why they are called sand dollar, like I just mentioned. They do have spines, but those spines compared to sea urchin spines are extremely short, right? And you can rather feel it. And if you actually closely look at in my pictures, you can see it. But if I give you a sand dollar, you can certainly feel it. They use their spines, which are also flexible, to burrow into the soft sand. So these guys actually, burrow like that, with where they actually kind of part of their discs are embedded in the substrate. Most sea urchins are herbivorous. They graze on algae, and they are voracious grazers. They are very fast in feeding into these algae and aquatic weeds or seaweeds. Sand dollars, on the other hand, are suspension feeders, right? They do have a particular structure, really cool structure called Aristotle's lantern. This is actually Aristotle's lantern. You're looking at a sea urchin. When you break up the sea urchin shell, remember what I mentioned about the fact that they have an endoskeleton. If you their endoskeleton is kind of fused. If you break it up, inside of that you see the Aristotle's lantern, right? Now I do not want you to be able to memorize this labeling. I provided that for you to understand what the heck this is, right? So it's called Aristotle's lantern because it looks like a lantern. It does not do anything like a lantern. Its purpose is to help the animal grind vegetation material they eat. Remember, vegetation material contains cellulose, and breaking cellulose is actually hard, so you need some very rigid, robust structures for the purpose of grinding, right? So there are this particular structure called Aristotle lanterns has muscles and surfaces, right, or teeth to do the grinding function. All right, let's move on. So like in generally about any echinoderms, sexual reproduction predominates in echinoids as well. Fertilization happens externally. Sexes are separate, males and females. Larvae has a long lifespan, and the larvae, interestingly, can actually, because they have a long lifespan, they can disperse from the size of fertilization through elsewhere, and them being planktonic, when the larvae being able to freely float with the water current helps this long distance dispersal. And in fact, like if you wondered where the tube feeds of these guys are, right? In this guy, you can kind of see the tube feeds are kind of located around the periphery, right? And what you cannot see is tube feed can also occur, right, in these areas, right? Um, as well, but it's not clear in this picture, right? Um, in this one, the longer ones are spines, and in between the spines, if you see these things that are protruding outside, right? These parts, those are tube feeds. So tube feeds can actually, can be extended. Like I said, it looks like a worm attached to the animal, right? So tube feeds are much more flexible than spines, right? In this one, these are actually the long spines, right, that I was referring to, right? All right. Okay. That's actually all I'm going to talk about echinoderms. I'm not going to talk about every group. I just wanted to talk about the most important groups within echinoderms. Right. Hey, if you want to know more, just ask me. All right. Let's now talk about chordates. That is an incredibly important group to discuss about. What are the key characteristics of a chordate? They